Okay. Very good afternoon to all the participants, and we can start with our plenary talk now. Good afternoon, Bin, and I'll give you a short introduction, and then you can start your presentation. We have with us Professor Chow Bin. He is an associate professor in the School of Civil and Environmental Engineering and a PI in the Singapore Center for Environmental Life Sciences Engineering, CELSI, Nanyang Technological University, Singapore. He obtained his PhD in Chemical and Biomolecular Engineering from National University of Singapore in 2008, and then he worked as a postdoctoral research fellow in Washington State University and the Pacific Northwest National Laboratory. Since he joined NTU in December 2022, he joined NTU in 2012. His research efforts have been focused on environmental biofilm engineering with the goal of understanding environmental biofilm processes and applying the insights to harness the power of beneficial biofilms and to combat detrimental biofilms in the context of environmental biotechnology. He currently serves as an associate editor for critical reviews in biotechnology, environmental research, water research, and frontiers in cellular infection microbiology in the biofilm section, as well as an editorial board member for Environmental International. So we are really honored to have Ben with us today, and he'll be talking about biofilm engineering for biotechnological application. He is an environmental engineering and he specializes in biofilm. So we will get a very nice insight into his work. And with this, I would like to request Ben to start his presentation.
Thank you very much. Thank you, Annie, for inviting me to give a talk here. And I'm very glad to have this opportunity, actually. I haven't seen you for quite a long time. Good to see you again. So I'm very honored to be here to share some of our recent work in the biofilm engineering. So now you can see my slides. Is that the presentation mode, or you can see the
No, it's in presentation mode.
So it's not a problem, huh? So it works.
No, it's fine.
Great. Okay, so I'm going to talk about biofilm engineering for biotechnological applications. But before I start, I would like to thank my team members. And it's because of them I have all the interesting results that I can share with all of you. And so biofilm engineering, of course, the first thing we want to talk about is actually what is a biofilm. So I've been working on biofilms for more than 10 years, but when we talk about biofilm, I still do not know how to define a biofilm. But most of us, we can use a very simple description, that is the aggregates of microorganisms that are embedded in the self-produced matrix of extracellular polymeric substances. And then we call them a biofilm. But nowadays, we know, it is not that simple. Like you have a few of cells that aggregate together, or you have 1,000 cells or 10,000 cells aggregate together. Which one will become a biofilm? So now we realize actually a very important aspect for biofilm, that is the emergent properties. So that means the biofilm is a lot more than a simple collection of individual cells. So the biofilm activates certain properties that are not shown by the individual cells. For example, biofilm, they have a higher tolerance to biocides, including antibiotics. And then there are some chemical gradients inside the biofilms. These are what we call the emergent properties. So if you look at the history of biofilm, and actually as early as in 1975, researchers start to use the term biofilm to describe a microbial film in a trickling filter. And this specific work, published in 1975, they look at the trickling filter samples, and then they observe the thin layer of microbial film on those pebbles, and they call it biofilm. So that's the starting point of the term of biofilm. And nowadays we all use the term biofilm to describe the films of microorganisms on the surfaces or interfaces. Now in environmental engineering, we have so many different types of biofilms. So for example, the river biofilms, these are actually the biofilms at the river sediment. And then we have the biofilms in wastewater treatment, and they help us to purify water to remove the nutrients from our wastewater. And we also have biofilms that are detrimental to our infrastructure, or they are not that good to us. For example, these are the biofilms growing in the water purification membranes, and this is what we call biofouling, and they compromise the performance of some membrane filtration. And then we also have biofilm growing inside the water distribution systems. This is a pipe that actually distribute water to the households. You can see a thick layer of biofilms inside, and these are the biofilms may cause potential problems to us. So in my group, I'm focusing on biofilm engineering. So what does biofilm engineering mean? And this term, biofilm engineering, was used as early as 1990s. At the time, when the Montana State University, they got the funding from the National Science Foundation to set up a center. It's called Center for Biofilm Engineering, so that's called CBE. And in that center, they define broadly biofilm engineering as the fundamental and applied biofilm research driven by industrial, environmental, and health issues. So basically, any issues that relevant to industries, environmental and health aspects, no matter whether they are fundamental or applied, they call them biofilm engineering. But actually, in 2017, the UK National Biofilms Innovation Center organized a workshop to look at the biofilm engineering, and they proposed 4 measures to intervene with the biofilm issues, including prevent, detect, manage, and engineer. So in this workshop, they clearly define engineer. So this specifically, they define the control and direct the complex microbial communities in process applications. So now a general consensus like biofilm engineering refers to the harnessing the beneficial uses of microbial biofilms, including populations or communities, by understanding the fundamentals of biofilm development process. So in my group, I've been focusing on 2 main aspects. Number 1 is to understand the biofilm bioprocesses in natural and engineered environments. The other aspect is to develop and advance biofilm engineering enabled microbial biotechnology for the environmental applications, for example, recovering of resources or removing of environmental contaminants, or for chemical synthesis, like producing biomaterials or biomolecules. So today I'm going to focus on the second aspect, that's for the biotechnological application part. I'm going to give you a few examples on how we engineer biofilms for the biotechnological applications. Whenever we talk about biofilm, and biofilm matrix is really important feature of a biofilm, and it is a self-secreted extracellular polymeric substances that consist about more than 80% of the biofilm biomass, and naturally they are absorptive. So that means they are able to absorb metals or organic compounds from the environment. Now, the key matrix components include the proteins, polysaccharides, lipids, and nucleic acids. Now, for us, we are looking at is it possible that we actually engineer the biofilm matrix so that we make the sorptive property a lot more specific? Although the biofilm matrix, they are naturally sorptive, the adsorption is not non-specific. So that means they can adsorb many different metals and many different organic compounds. They do not have much selectivity at all. So now, if we look at the matrix components, one very important component is proteins. And proteins are something that we can manipulate. So that means that we are able to genetically engineer the matrix so that we can change the component of proteins inside the matrix and make the biofilm matrix based adsorption a lot more specific. So the idea is, are we able to convert a biofilm into a programmable biomaterial for resource recovery and the contaminant removal? So we start with one very, it's a kind of global issue. That is that's phosphorus issue. So we have the very big contradiction in phosphorus in the environment. So on one hand, we have we are lacking of the phosphorus for food production. On the other hand, there are a lot of phosphorus that going into our environment, especially the aquatic environment, and that cause big problems like eutrophication. And how to solve the problem? Of course, nowadays we have the wastewater treatment plants. We're trying to recover phosphorus from the wastewater, and the mostly use the process called EBPR that's enhanced the biological phosphorus removal, but the effluent P level is still relatively high, and they may also cause problem and also actually disperse phosphorus to our environment. So we were thinking, is it possible for us to engineer a biofilm and makes it a lot more specific and highly specific for phosphorus, so that we can recover phosphorus from dilute solution. So that means you have a solution containing very low level of phosphorus, but we are able to recover phosphorus from there. So how to do that? The idea that we have is actually we are looking for the bacteria systems. So bacteria, they grow in the environment, and in the environment, phosphorus is usually the limiting nutrient. So the bacteria have evolved, and they have very strong capability in capturing phosphorus from the environment. And one system is these we call PSTS, that's phosphorus binding protein. And this protein has 14 hydrogen bonds that can bind to phosphate. So it is very strong binding to phosphate, and it's also very specific to phosphate. So now we are. So the idea is it possible for us to display this phosphorus binding protein into a biofilm matrix, so that the biofilm, you have bacterial cells and the matrix. Now inside the matrix, you have a lot of these PSTS protein there. That's phosphate binding proteins inside. And then eventually, and if we grow the biofilm in a reactor, and we have the P-rich solution going in there, and then all the P inside the solution, or most of them, will be captured by the biofilm. And then eventually, and because of the binding is through hydrogen bond, and if we change the pH, the hydrogen bond will be broken, and then phosphate will be recovered. So that's the general idea. And then we try to find a host that we can use to test it. So we used a bacteria that's called Shrinella amiodensis, and this is a environmental bacterium. And we use this because the biofilm matrix of this bacteria has already been identified. So one of the very abundant proteins called BPFA inside this bacteria, and this is one of the most abundant protein, and it's an ideal vector for us actually to modifying biofilm matrix. And we had done some previous work actually to characterize the EPAS of this biofilm, and we have also had some work. We changed the composition of the matrix and make it useful for some other purposes. Now, so what we want to do is, are we able to display PSTS inside the biofilm matrix for phosphate recovery? And this is the method we use. This is actually a homologous recombination. And what we do is actually BPFA is the most abundant matrix protein inside this biofilm. Now the idea is, are we able to link PSTS to BPFA so that whenever you have BPFA inside the biofilm matrix, you will have PSTS at the same time inside the matrix. And then eventually, because BPFA is one of the most abundant proteins in the matrix, and eventually your PSTS will be abundant too inside the biofilm matrix. And then your biofilm become biofilm matrix contains PSTS that is very specific for phosphate binding. And this is some of the data that we have at a neutral pH, and we have the Y type that's non-specific binding to phosphate, and then we have the mutant that has PSTS displayed inside the matrix, and actually we have a significantly more phosphate can be adsorbed by this biofilm. But at a higher pH and a non-specific binding we can observe there. So that means most likely we can change the pH and recover phosphate from it. And this is based on Manisha's work, and we haven't published it yet. And then at the same time, we were thinking, now we are able to recover phosphate from the dilute solution using the biofilm. So we were thought like, is it possible actually for us to use a similar strategy to remove some low concentration contaminants from water? And then we look at the environmental issues, and we found in Southeast Asia, and especially in South Asia, we have a big problem that is called the arsenic contamination problem. Right, so arsenic contamination in groundwater has affected a lot of population. Now, if you look at Asia itself, around 120 million people are affected by arsenic contamination in their drinking, in their groundwater. Now, that's about 60% of the total population affected globally. So in rural areas, like for example in the Nepal or the Bangladesh, for example, and in those villages, what they use is actually something like this: is sand columns, and then actually the water pump out, and then they filter through the sand column, and then they rely on the sand column to remove arsenic. But actually that's not very effective in most cases, because this removal highly dependent on the iron to arsenic ratio. So you need to have a lot of iron in your system so that it will work. That's based on chemistry. Now our idea is actually, can we engineer the biofilm inside the filter, inside the sand filter? If we can grow a biofilm inside the sand filter, that biofilm can help us to specifically remove arsenic from water, and that will be very useful. So that is the ASEAN's work, and what we did is actually we are looking for an arsenic binding protein, and we screened for arsenic binding protein from different organisms, and we look at the arsenic binding motifs, and then we eventually choose one from E. coli, and then we design these. What we want is actually we display this ARSR inside the biofilm matrix. So I design a biofilm matrix that can be used for arsenic sorption and with a very high specificity. And this is the general idea as what we have done for the PSTS. So for PSTS is a BPFA in the chromosome, the link to PSTS, that's for phosphate binding. Now what we can do is replace PSTS with ARSR, and then eventually we have BPFA linked to ARSR. So in the biofilm matrix, whenever you have BPFA, you will have ARSR, and then the biofilm matrix is able to remove the arsenic. But the problem here is actually because this integration is inside the chromosome, so the copy number is 1, and also it's not that controllable for us. And what we want is, are we able to make it controllable using a plasmid expression system, so that whenever we are able to change the expression level of this BPFA linked to ARSR. So the idea is very simple, but the problem here is actually BPFA is really, really big. So it's itself is already more than 8 kb. So it's very big, and we tried a few times and actually failed. Now, so how to solve the problem? And we discussed this, and then eventually found a way. So are we able to truncate BPFA into different pieces? And then we just use parts of the BPFA. And then we link to ARSR whether they work or not. So we are using truncation method. So what we do, if we look at BPFA, you have different domains. Now we have we use 4 different designs. We just keep one or two domains inside BPFA. And then eventually we found two of them does not work because that will affect the biofilm formation greatly. So if they cannot form biofilm, of course, we do not want to use. And then we use the other two truncation configuration, and then we link to ARSR, and this is what we get. So this is the white type. You have the arsenic binding, that's non-specific binding. And then we have the white type plus the chromosomally encoded ARSR. So we are able to remove some of the arsenic from the water. But if we're using the plasmid-based removal system, and we get a lot higher amount of arsenic to be removed, and it's very specific too. So this is some of the some of the data that we have. We're preparing the manuscripts now. And then that's for the environmental biotechnological applications. But on the second aspect, can we engineer biofilm for biomanufacturing? So, I believe most of you have already known that actually a few months back, President Biden in the US, they announced the launching of the National Biotechnology and Biomanufacturing Initiative. And the biomanufacturing is actually use biological systems to produce commercially important biomolecules. So that can be used for agricultural purposes, food, materials, energy, and the pharmaceutical industries. So it has been estimated that around 30 trillion that in terms of the total value of biomanufacturing by the end of the decades. So a huge market there. Now, if you look at the biomanufacturing, so we start from biomanufacturing 1.0, that's many years ago. That's just based on the primary metabolites from fermentation. For example, acetic acid fermentation or alcohol fermentation. That is a 1.0 generation. Now, then we have the 2.0 generation, like we are producing some secondary metabolites, for example, antibiotics, for example. And then the third generation to produce a large size biomolecules that combine with a recombinant DNA technology and advanced cell culture, for example, antibody production, for example, and enzyme production. Now, we are entering the fourth generation biomanufacturing to produce new products that to meet the grand challenges that we are facing now. So that will combine metabolic engineering, synthetic biology, precision fermentation all together. Now, why do we need biofilm for biomanufacturing? And I'm going to use a simple example to explain that. Now, in this case, we are trying to convert indole into tryptophan. And this is very simple. You just need to place two genes into E. coli, and then E. coli is able to convert indole to tryptophan. Now, if we look at the culture, this is a planktonic culture for the production of tryptophan. But after some time, you see tryptophan concentration decrease. At the same time, your indole increases. So that means in planktonic culture, you can observe the reverse reaction. After some time, you will see the reverse reaction already. Why? That's because in the planktonic culture, especially in the batch mode, you have all the products that are accumulated in the reactor. So if the product concentration is high enough, and then you will see the reverse reaction. Now, if we can use biofilm, and then biofilm-based production, we are able to continuously remove the product from the system, and the catalysts are still there because they are biofilm, they are adherent to surfaces inside the reactor. So in that case, we are able to prevent the reverse reaction. So this is the idea. Now, if we grow the biofilm and we use the biofilm to convert indole to tryptophan, if it is planktonic culture, we observe the reverse reaction. But if we are using biofilm for this catalysis, we are able to address this reverse reaction problem. But at the same time, we are creating a new challenge That is the stability of the biocatalyst. Because the biofilm as a catalyst, the biggest challenge is actually how to control the long-term stability of the biocatalyst. Because the biofilm is actually your cells, they form a three-dimensional structure, and they are, so you will see some cells that move away from the biofilm, especially in the presence of certain chemicals. Now, that's the biofilm dispersal. Now, how are we going to address this? So what we do is actually we are using light to enhance the biofilm stability. So we use NIR, that's near infrared light. And whenever we have this light exposure, and this gene, BPHS, this specific gene can respond to light. Now, whenever we have this NIR light, and we will produce more cyclic diGMP. This is the signaling molecule in biofilm formation. If you have a higher C-diGMP level, you will form a more coherent, more stable biofilms. So in that case, we engineer the biofilm so that in the presence of NIR light, we will keep the biocatalyst very stable, and eventually we are able to increase the yield by 30%. That's the benefit of using biofilm for biomanufacturing. But the problem here is actually it's not like the more biofilm you have, the better. Why? That's because whenever you have biofilm there, you have to face the solute diffusion problem. So the solute diffusion problem is inside the biofilm, the diffusion is a lot lower than those chemicals diffusion in water. And if you look at a simple example, if we have a biofilm here, now if the biofilm is thick enough, and then eventually your substrate diffusion is going to be a problem, especially to the bottom of the biofilm. Even for oxygen, then the diffusion may be a problem if your biofilm is thicker than 40 micrometer. So if you have a very thick biofilm, eventually you will have a lot of unproductive biomass at the bottom. So that is not what we want. And so we want to have a very productive biofilm. And so we want to prevent the thick layer of unproductive biomass. So how to control the biofilm thickness? So the approach that we use is to using cyclic diGMP based controlling method. So as what I mentioned, the higher the diGMP is, the more biofilm you will have and the more stable they will be. So now if you reduce diGMP and the bacterial cell will like to move away from the biofilm and become planktonic again. So if, what we need is just to control cyclic diGMP level. How to control that level? You have DGC to synthesize diGMP. We also have PDE to degrade diGMP, and we just need to control the enzymes. So what we need is to control DGC and PDE so that we are able to control diGMP. And then eventually, your biofilm dynamics is a function of diGMP. Then we are able to control the biofilm. And the method that we use is what I mentioned just now, is a near infrared light responsive protein. And this protein is able to synthesize the diGMP in the presence of a near infrared light. And this is the design. Because in our light increase the diGMP level, so you increase the biofilm formation. But if the biofilm is too thick, we have to find a way to reduce the diGMP level. So we use the blue light responsive PDE, EB1. So this is specific gene actually respond to blue light. And when you have blue light, your PDE activity will be activated, and then your diGMP will be degraded. So in that case, what we achieve is actually, if we have a red light, the NIR light, your biofilm will be thicker. And then if we change the blue light, the biofilm will become thinner. And then eventually what we can do is just to use alternate exposure to red light and blue light. We are able to control the thickness of the biofilm. And then we demonstrate this approach by Manisha and Yidan. So we are able to control, so we combine the light controllable, this is the increase of biofilm formation part, this is decrease of diGMP part, and this is a chrome quenching part. And then eventually we demonstrate that we are able to use light to control the dynamics of the biofilm, and then we use this quantum quenching biofilm for biofouling control. So this is just a simple example. If you are interested, you can read our papers. I'm not going to talk about details of this. And then eventually what we want, are we able to use this light controllable gene circuit together with the precision fermentation? And this is the work that we have done a few years ago. And what we did is actually to synthesize FMN. This is a flavin mononucleotide. This is very important in food industry and also in pharmaceutical industry. And we engineer the bacteria to produce FMN. Now the idea is that is it possible for us to combine with the red light and blue light controlling dynamics, so that we just need to change red or blue light, we are able to change the biofilm thickness to achieve the optimal production of FMN. We failed. That's because the light controlling gene circuit is not optimal. BPHS and BPH, this is a red light responsive gene and the blue light responsive gene. And we are now engineer these two specific genes, try to make the whole gene circuit optimal. And then we are able to use this for the biofilm-based precision fermentation for chemical production. So in summary, I just show you a few examples of how we can engineer biofilm for biotechnological applications. And there are a lot of opportunities for us to do that. We have known a lot about biofilm biology, but how to use those biofilm biology insights to engineer the biofilm for some biotechnological applications. That's areas that we are exploring. And biofilm biology informs us for the platform technology development. And I show you the resource recovery and contaminant removal. There's two examples, and also show you a few one example on the continuous bioprocess for biomanufacturing. That's chemical production, and potentially special control. That's actually the dynamic of the biofilm for the biofilm-based biocatalysis. Thank you very much. I think I used 30 minutes.
Yes, perfectly.
Good, thank you, thank you.
Thank you, Vin. Thank you for sharing your interesting insights into the biofilm matrix and how that can be used as a catalytic, location for different types of application, like arsenic removal. And you talked about how the biofilm thickness can be controlled. So thank you so much. And now the floor is open for any questions that we have.